Today we'll be talking about Golgi tendon organ. It is also called as inverse myotactic reflex or inverse stretch reflex. It is also called as reciprocal activation or autogenic inhibition. We'll see why is it called so one by one. Now, this is a structure of a muscle and this is a bone. So what is the structure which is connecting, which is present in between or which is connecting muscle to the bone? Yes, it is the tendon. And inside the tendon, you can see this green color structure and blue color structure. So this blue color structure, they are the collagen fibrils. And inside the collagen fibrils, we have Golgi tendon organ. Golgi tendon organ, what do we deduct? It is going to deduct increase in the tension. So whenever, let us say you have gone to the gym and you are into the beast mode and you are not supposed to lift so much of weight, but because you have gone into the beast mode, you keep on lifting weight higher, more and more, and suddenly the tendon have chances of ripping off from the bone. That is called as tendon avulsion. So whenever there is increase in the tension or overstretch of a tendon, this GTO, that is Golgi tendon organ, is going to be activated. And what are the sensory afferent? It is type 1B fibers. So afferent are type 1B fibers. So this afferent is going to go to the center, that is to the central nervous system or spinal cord. So whenever muscle is contracted, so there is increase in the tension develop, it is going to stimulate our Golgi tendon organ. So this I have made an image. It is a cranky image because there is increase in the tension. So whenever GTO is stimulated, the afferent is carried via the type 1B fibers. So now if I magnify the sensory neuron, so whenever there is any increase in the tension developed, so this is a figure of a type 1B neuron. So there's going to be increase in the tension is going to cause opening of mechanically gated ion channel. So there is going to be influx of the sodium. So there's going to be influx of sodium. So lot of positive ion is entering. So whenever lot of positive ions is entering, so resting membrane potential, it goes and it touches the threshold potential. So when it touches the threshold potential, this is an axon. So it is going to open voltage gated ion channels. So increase in the tension, there's going to be opening of mechanically gated ion channels, sodium channels. So there is lot of entry of positive ions, which is going to increase our resting membrane potential to threshold potential, which causes opening of voltage gated ion channels. And further, there will be influx of sodium ion. And this information will be going to our spinal cord. So whenever muscle is contracted, increase in the tension generated, this will go via the sensory afferent, that is type 1B fibers. So because it is sensory, the cell bodies are in the dorsal root ganglia. Now it is go and it is going to enter again because it is sensory. It is going to enter the spinal cord. The fibers are going to enter the spinal cord via the dorsal horn because dorsal horn is sensory. And after the sensory afferents enter the spinal cord, here it is going to bifurcate. It is going to bifurcate on two interneurons. And this interneurons, they are going to go and synapse on the motor neurons. Now, which interneuron is inhibitory, which is excitatory? It depends on the amount, it depends on the type of neurotransmitter which is secreted. If the neurotransmitter which is secreted happens to be an inhibitory neurotransmitter, then this interneuron will be inhibited. If the neurotransmitter which is released is excitatory, then this neuron will be stimulated. So whenever there is increase in the tension, the Golgi tendon organ is going to sense this increase in the tension, and the afferent is via the type 1B fibers which go via the dorsal root ganglion, enter the dorsal horn of the spinal cord, and it is going to bifurcate the interneuron. And then you tell me if you want the agonist muscle uh, to because there is increase in the tension, so we want the agonist muscle to be inhibited or to relax. So which type of interneuron you want to go, you want it to go and synapse with the motor neuron? Obviously the one which is having inhibitory neurotransmitter. So this interneuron will go and will synapse on the alpha motor neuron, and it is going to go and inhibit or relax the agonist muscle. Now, if you want agonist muscle to relax, the antagonist muscle should contract, obviously. So this is called as, then if you want the antagonist muscle to contract, the interneuron, which is excitatory in nature, it is going to go and it is going to supply the motor efferent is going to go, alpha motor neuron is going to supply the agonist muscle. So here what is happening, there is reciprocal activation because you want agonist muscle to be inhibited or relaxed. So obviously the antagonist muscle should contract for agonist muscle to relax. This is called as reciprocal activation, and because there is inhibition of the agonist muscle, we call it as anti-autogenic inhibition. So Golgi tendon organ is also called as inverse because this is opposite of the stretch reflex. Stretch reflex is also called as myotactic reflex. So we call this as inverse stretch reflex or inverse myotactic reflex. Why it is called reciprocal activation? Because it is causing contraction of the antagonist muscle. Why is it called as autogenic inhibition? Because it is causing inhibition or relaxation of the agonist muscle. So, whenever you are going to hit the muscle with the knee hammer, then increase in the tension is detected by our GTO. This information is sensed by type 1B fibers because the afferent to the GTO is the type 1B fibers. The afferent from the GTO is the type 1B fibers. And then this type of fibrin, the afferent will go via the dorsal horn and it bifurcates and it synapses on two interneuron. And because we want the agonist muscle to relax, so the inhibitory interneuron will go synapse on the efferent neuron, that is alpha motor neuron, will go and inhibit the agonist muscle, and to contract the antagonist muscle, the excitatory interneuron will synapse on the motor efferent and will go and contract the effector. So this sensory receptors, the proprioceptors, then the afferent, the center, the efferents and the effector. This is called as a reflex arc. So what is the type of interneuron or if the neurotransmitter which is released by the interneuron happens to be a glycine, so IPSP, that is inhibitory postsynaptic potential will be generated, that interneuron will be inhibitory because glycine is an inhibitory neurotransmitter. And if the neurotransmitter which is released is glutamate, and glutamate being an excitatory neurotransmitter, so we'll have excitatory postsynaptic potential EPSP generated at the postsynaptic membrane. So this is an example of a Golgi tendon organ of the lower limb. So whenever there is an increase in tension, uh, the afferent will go via the type 1B fibers. It is going to enter the spinal cord. It is going to bifurcate and synapse on two interneuron. So outcome is we want the agonist muscle to be inhibited. We want autogenic inhibition and reciprocal activation. So these are the efferent fiber to the muscle associated with the stretch tendon. Okay, and this is the efferent fiber. The pink color one is the efferent fiber to the antagonist muscle. Here plus means excitatory synapse and minus means inhibitory synapse. Now, the Golgi tendon organ, it is the opposite of the stretch reflex. Contracting the muscle activates the Golgi tendon organ. Afferent Golgi tendon neurons are stimulated. The neuron inhibits the contracting muscle and the antagonist muscle is activated. Hence the name reciprocal activation or autogenic inhibition. As a result, the contracting muscle relaxes and the antagonist muscle contracts. So if you tap on the tendon of a muscle, it is going to contract, its synergist contract and its antagonist are inhibited. It is an example of a polysynaptic reflex. So a tap on the patellar tendon, it is going to stretch the extensor muscles and its sphincter. So here initial stimulus is muscle stretch. Okay, this is muscle sphincter. The afferent impulses from the stretch receptors will go to the spinal cord. Here it is going to bifurcate on the interneuron, and this efferent impulses to the alpha motor neuron will cause contraction of the stress muscle that resists or reverses the stretch. And this is the efferent impulse to the antagonist muscle are dampened. This is reciprocal inhibition. Okay. So what is reflex physiology? Reflex are automatic, subconscious response to change within or outside the body. Reflexes, they maintain the homeostasis, that is autonomic reflexes, heart rate, such as heart rate, breathing rate, blood pressure, and digestion. Reflexes, they also carry out the automatic action of swallowing, sneezing, coughing, and vomiting. Reflexes, they maintain balance and posture. Examples, the spinal reflex, which control the trunk and the limb muscle. And we have brain reflexes. They involve reflex center in the brainstem. Example, reflexes for eye movement. So we have seen a reflex arc. It governs, what is a reflex arc? It governs the operation of the reflexes. So nerve impulses, they follow nerve pathways as they travel through the nervous system. The simplest of these pathways, including a few neurons, constitutes a reflex arc. So reflexes whose arc pass through the spinal cord, they are called as spinal reflexes. So what are the components of a reflex arc? First is the receptor. Second is the sensory neuron or the afferent. Third is the integrating center. The fourth is the efferent neuron or the motor neuron. And the fifth one are the effectors. So parts of a reflex are the receptor. What is the role of a receptor? It is going to detect the stimulus. So description, the receptor end of a particular dendrite or a specialized receptor cell in a sensory organ. And what is the function? Sensitive to a specific type of internal or external change. Then we have sensory neuron. The second component of a reflex is a sensory neuron. It conveys the sensory information to the brain of the spinal cord. Description, the sensory neuron could be a dendrite. The information can be carried from the dendrite, from the cell body, or from the axon of a sensory neuron. And the function is it is going to transmit nerve impulses from the receptor into the brain and the spinal cord, that is towards the center. Then third component of a reflex axis is interneuron. It is going to relay the neurons. So interneuron, it could be a dendrite, cell body, and an axon of a neuron within the brain or the spinal cord. The function of an interneuron, it serves as a processing center, conducts nerve impulses from the sensory neuron to a motor neuron. And the fourth component of a reflex axis is motor neuron. The function is its conduct motor output to the periphery, that is transmit the nerve impulse from the brain or spinal cord to an effector. Description, motor neuron can be a dendrite, cell body, and an axon of a motor neuron. Now this is the flowchart showing the stress reflex and the inverse stress reflex. So inverse stress reflex, which is the topic of our concern today. So what is the stimulus for inverse stress reflex or Golgi tendon organ or inverse myotactic reflex? Overstretch. So whenever there is increase in the tension or overstretch, or whenever there is opportunity of tendon avulsion, the GTO Golgi tendon organ will be activated. So stimulus is overstretch. The receptor is Golgi tendon organ. The afferent is 1B. And center is in the spinal cord. The efferent will be inhibitory interneuron, which is going to go to the effectors. Effectors alpha motor neuron, the extraphyseal fibers causing muscle relaxation. Because relaxation is happening here, we call this as lengthening reactions. You can remember this is another name for inverse stretch reflex, and there is reciprocal excitation. So same side I have kept stretch reflex so that you can compare and contrast the stretch reflex and the inverse stretch reflex. Anyways, this I have shown you in my stretch reflex class. So because today our topic is GTO, so I just want you to look at the flowchart of the inverse stretch reflex. Keep both side by side, compare and contrast. Okay. So it is also inverse stretch reflex, also called as autogenic inhibition or reciprocal activation. So this is a figure showing the muscle spindle, stretch reflex, muscle spindle, afferent is 1A, spinal cord. There is a transmitter release, neurotransmitter is a glutamate, efferent alpha motor neuron, extraphyseal fiber leading to contraction of the muscle. And this is inverse stretch reflex, overstretch or excess tension. The GTO will be stimulated. The afferent will go via the type 1B fibers. It is going to go to the spinal cord. It's going to bifurcate and synapse on interneuron, inhibitory interneuron glycine. Then the efferent or alpha motor neuron, it is going to go to the extraphyseal fiber and it is going to cause reciprocal activation or excitation of the antagonist muscle. This is happening to prevent tendon avulsion. Okay, these are the other types of spinal cord reflexes, that is withdrawal reflex. So whenever you are keeping a leg on a painful stimulus, it is going to activate nociceptors, the primary sensory neuron. It is going to enter the spinal cord and it is going to diverge. One collateral will activate ascending pathway for sensation of pain and postural adjustment. So there will be shift in the center of gravity. So withdrawal reflex is going to pull the foot away from the painful stimulus. This is a crossed extensor reflex and it is happening to support the body as weight is shifting away from the painful stimulus. So same side there will be contraction of the flexors muscle and extensor will be inhibited. On the opposite legs, extensors will be contracted as weight is shifting to the left leg and flexors will be inhibited. Crossed extensor reflex, it is a it's a polysynaptic reflex and the stimulus being painful stimulation of one foot which causes flexion, that is withdrawal of the ipsilateral leg as well as extension of the contralateral leg to stabilize the posture. Thus the same side leg flexors are activated and the extensors are inhibited and opposite thing and vice versa in the contralateral leg. Thank you.